Hello guys, hello family, hello friends. This is Dr. T. Welcome to Clinical Medicine. So guys, in this video I want us to do part 2 of our, remember we did discuss the basics, biochemistry of diabetes, biochemistry of diabetes. The biochemistry of diabetes. So guys, please watch, make sure that you watch part 1 before you come to part 2. Just to recap, so we said that, so this is our stomach firstly, and this is our, I'm gonna zoom into the enterocytes. So the enterocytes are the epithelial cells that line the intestine or the intestines. So this would be one enterocyte, the second enterocyte, another enterocyte, right? And we do know that on the other side we've got the blood vessel, a capillary, right? So this is our lumen. If you have watched part 1, there's a part where we talk about the classification of carbohydrates, monosaccharides, disaccharides, and the polymers. So whatever that we eat is complex carbohydrates, but we want to break everything down to the simplest sugars, which is galactose, glucose, fructose or fructose. So now we have got our glucose molecules, our galactose molecules, our fructose molecules there. So in part 1 I do discuss in detail, I do get into the details of how do you get glucose to enter the enterocytes. That's what is absorption. So glucose enters the enterocytes through special proteins called glucose transporters. Glucose transporters. Glucose transporters, there are two types. There is sodium glucose linked transporter, and there is facilitated diffusion glucose transporters. This is GLUT. So as I explained in part 1, the sodium glucose transporter has got two isoforms. There's SGLT1, SGLT2. SGLT1 is the one that is responsible for the absorption of glucose from the intestinal lumen into the enterocytes. It's this one. It's this one. So there are many of them on the apical membrane. So they absorb glucose into the enterocytes, right? But we don't want glucose to be in the enterocytes. We want glucose to be in the blood. So how does glucose move within the enterocytes and exit on the basal membrane? That job is done by the GLUT transporters. There are 3 classes: class 1, class 2, class 3. But here I'm going to specifically talk about GLUT 1, GLUT 2, GLUT 3, and GLUT 4. So glucose enters on the apical membrane through the sodium glucose transporter 1, but exits on the basal membrane through the GLUT1. That's how glucose enters and it gets deposited or enters into the blood. So after we've eaten whatever that we eat, talking about carbohydrates, it's broken down, it's absorbed, gets into the blood. Now we've got a state of postprandial hyperglycemia. Right? So we've got a lot of glucose that is here now. So hyperglycemia, there's nothing wrong with hyperglycemia. Everyone gets hyperglycemia. When you get hyperglycemia, it means that you're actually absorbing glucose from the intestine. That's good. But what is not good is a chronic state of hyperglycemia. That's not good, because that's how endothelial cells and organs, they get to be damaged. They get to be damaged by a chronic state of hyperglycemia. Hyperglycemia for 2 hours, hyperglycemia for 3 hours will not do anything. So now after we've eaten, this is what we get: hyperglycemia. But we want our glucose to enter the cells. That's where it's wanted. The brains, they want it. The muscles, they want it. Adipose, they want it. And all these different tissues, they want the glucose. The heart, they want the glucose. So we cannot just have glucose in the blood. That's not where we want it. We want it to be in the cells. How does that happen? Okay, so we said, let me just make sure that I label. So this glucose transporter here is the sodium glucose 1. Sodium glucose linked transporter 1. Then this one here is the GLUT glucose transporter 1. Right? So just create space. Let me just finish this one. So where do we get glucose? Okay, where do we get SGLT1, sodium glucose linked glucose transporter? One is in the enterocytes, the apical membrane of the enterocytes. The apical membrane is the membrane or the side of the enterocyte that is facing the lumen. And SGLT2 is found in the kidneys, specifically the proximal tubule. Glucose transporter 1 is found in the basal membrane of the enterocytes. But the basal membrane. Glucose transporter 2 is found in the beta cells of the islets of Langerhans, in the pancreas, obviously, and also in the hepatocytes. Very important. This is very high yield stuff. And then glucose transporter 3 is found in the brain. And then glucose transporter 4 is found in the adipose, fat tissue, and in the muscle. Very important, guys, very important, because we are going to talk about those later on. Sorry. So now let's move forward knowing this information. All right. So now, if you watch part 1 of Biochemistry of Diabetes, I explain exactly how this function. how this pump actually, this co-transporter, how it works, because it uses an ATPase, which is on the basal membrane. So I explained that. So please go and watch that video. Okay. So guys, so let's say now I've just had 2 liters of Coke and a loaf of bread, and I've done my digestion, my absorption. Now I've got hyperglycemia. That's where I'm at now. So what's going to happen? What's going to happen is that this glucose is going to be transported in the blood. It's going to be in the blood, and it's going to be transported to the pancreas. So specifically the islets of Langerhans, and more specifically the beta cell. So let's take one beta cell and enlarge it. So these glucose molecules are gonna go and enter the beta cells of the islets of Langerhans. They will enter via a glucose transporter that we spoke about, which is the GLUT2. So they enter through the GLUT2 transporter. Once glucose enters the beta cell, glycolysis takes place, and ATP is produced. And so basically hyperglycemia stimulates some kind of chemical reactions within the cell, in the cytoplasm. So I'm not going to get into the details of how all of that happens, but eventually what happens is that there's an influx of calcium. Once there's a calcium influx, remember inside these betas, the main function of beta cells is to synthesize and store insulin in vesicles or granules. So now we've got insulin inside. So I'm going to represent insulin with this orange marker. So once there's a calcium influx because of the glycolysis that occurs, because of the influx of glucose through the glucose transporter, the vesicles, they move towards the plasma membrane of the beta cell, and endocytosis of insulin occurs. So now insulin, which is a protein, gets released into the blood now. Remember, it's a polypeptide. So that insulin is going to get into the blood. What will it do there? So this is how insulin is released. So this is how the beta cells sense the state of hyperglycemia through the GLUT2. The GLUT2s are very important for sensing hyperglycemia, because they're the ones that allow glucose to come in, and then because there's a lot of glucose that comes in, there are chemical reactions that takes place in the cell. One of them is glycolysis, and then there's an influx of calcium, and then because of that influx of calcium, the cell depolarizes, and then the vesicles that contain insulin, they move towards the plasma membrane, and insulin is released through the process of exocytosis. Now what will that insulin do? Okay, so what other organ has got GLUT2? We said the hepatocytes, the liver, they also have got GLUT2. The hepatocytes, they also have GLUT2, which means glucose is able to freely enter the GLUT2 in both of these organs. What is special about GLUT2 is that it's insulin independent. Of insulin. So if it's insulin independent, it means that the GLUT2 opens to glucose in the absence of insulin. So glucose is able to enter the pancreas, the beta cells specifically, and the hepatocytes in the absence of insulin. So the uptake of glucose by the hepatocytes is not dependent on insulin. That's what I'm trying to get you guys to understand. Okay, so now the insulin, what is it gonna do? What's the role of insulin? We want insulin to help us with this state of hyperglycemia. We don't want this glucose to be high. We don't want these high levels of glucose in the blood. We want it to get into the cells. That's why we need the insulin. So, but what will insulin do? Okay. So, this is the pancreas. This is the liver. Now, let's draw the adipose. Adipose tissue. These are adipose cells. So the adipose cells, they've got GLUT4 transporters. They've got GLUT4 transporters. And also the muscles, they've got GLUT4 transporters. Even the muscles in our bodies, they've got GLUT4 transporters. Right? But these GLUT4 transporters, when there is no insulin, they are stored or they are kept inside these cells. They are kept inside. Once insulin comes and binds to its receptors, insulin receptors, so the V shape is trying to show insulin receptors. So the insulin will come and bind on these receptors. Once insulin binds on these receptors, there's a lot of intracellular chemical reactions that are going to happen, but eventually what's going to happen is that the GLUT4 transporters that are stored in the cytoplasm, they become expressed. So these are the GLUT4s. They become expressed on the cell membrane of these cells. They become expressed. Imagine having hypoglycemia and you express GLUT4. It means you're going to drop that blood sugar further, because let's say you've got a blood sugar of 4 and your GLUT4 are expressed. It means the little blood sugar that you have is going to move into the cells and you're going to have hypoglycemia. So the reason why GLUT4 transporters are stored within the cell are not expressed when there is hypoglycemia is because they worsen the hypoglycemia, because they allow the movement of glucose from the blood into the cell. So that's why you need insulin to bind to its insulin receptors so that the GLUT4s can be expressed. Once there is no insulin, these GLUT4 transporters, they are endocytosed. They get back into the cell again. So, okay, we're saying that insulin binds to its receptors on the cell surface of these two tissues. Once insulin does that, the GLUT4s that are stored within the cytoplasm of these cells becomes expressed on the outside of the cell. When that happens, now you get a massive movement of glucose into these tissues via those GLUT4 transporters. This is how adipose cells uptake glucose. This is how muscle tissues uptake glucose. They are dependent. So the GLUT4 transporters are insulin dependent. Without insulin, no GLUT4 is expressed and no GLUT4 is activated. So the GLUT4s are very potent when it comes to moving glucose into the cells. The GLUT4s, glucose transporter form. So I hope you guys get this, because that's where diabetes is, right? That's where it is. So once now you've got a massive movement of insulin, massive movement of glucose into these tissues via the blood for transporters, now that state of hyperglycemia is corrected. The glucose starts to drop in the blood now, and then you get to the normal glycemia. That's how glucose moves into the cells. Maybe you ask yourself, so we said that GLUT2 is found in the beta cells and also in the liver. The GLUT2 is not very potent in normalizing glucose. These are the ones that are very potent, and these are the ones that are implicated in diabetes, which is a chronic state of hyperglycemia, right? Okay. I'm gonna leave it here, guys, because I don't want us to have very long videos, but I'm gonna finish in part 3. Guys, please subscribe. Please subscribe.